here's a slide of something you might typically see in an undergraduate textbook. What we've got here is a baby, and the baby is really meant to represent a data cloud. And what it's showing are correlations that exist between different ages and skills that babies might have. So it's describing what babies can do at various ages. For example, at 6 months, well, they can pretty much roll over, and at 10 months, let's say, they can pull themselves up. But these are just correlations. What it's not doing is it's saying anything about how that development occurs. How is it that one level of competence gets transformed into the next level of competence? That is, what are the mechanisms and processes involved in development? And as developmental cognitive neuroscientists, what we're really interested in is understanding the causal mechanisms of behavior. We want to know what causes people to act on a moment-by-moment basis, but also what causes them to grow and develop on the kind of developmental timescale. Why is understanding mechanisms important? Well, because by understanding the mechanisms that underlie learning across different individuals, we can start to answer questions such as why does learning take place in a certain way and not in other ways? Or why does learning take place at certain times and not other times? So why does language kick in between, say, 1 and 2 years of age, no matter how much experience, broadly speaking, babies have? And why is some kind of learning quicker in some individuals and not individuals? Also, if we have an understanding of the mechanisms of how the brain changes and the learning involved in the brain, we can start to explain why and how certain neural systems break down and how to fix them. So here we have, for example, 3 examples of case studies that we would study in this module of children with autism, Down syndrome, and Williams syndrome. Now, the work in this module is delivered by the people at the Center for Brain and Cognitive Development, and this is really a world-leading expert center trying to understand the relationship between brain and cognitive development. They've really developed the methods, the cutting-edge methods that are being used all around the world now. And the mission of this center is really to investigate the relation between postnatal brain development and changes in perceptual, cognitive, and linguistic abilities across the lifespan. Now, the domain that does this is really what we call developmental cognitive neuroscience. And developmental cognitive neuroscience is a multidisciplinary approach that uses really 3 different types of methods to try to triangulate what the mechanisms underlying behavior might be. And I'm going to say a little bit about each of these methods. So the first one is really observation and experimentation. The next one is using the neural and biological constraints of the growing child as serious constraints on what might be going on. And the third one is computational modeling. So what are the observational and experimental methods that are used? Well, in developmental cognitive neuroscience, it's inherited an experimental perspective. So we use scientific methods. Studies are hypothesis-driven. We rely on observations to either verify or falsify the hypotheses that we put forward. What we're really interested in is generalizations that apply across the individuals. So one reverse side of that is that people tend to sweep away individual differences about you and I, but rather treat us as the same kind of person. We're trying to find out some truths that apply across the board. It's an approach that draws on quantitative analysis, so there's a lot of statistics that are applied in this field. And in terms of the kinds of tasks we do, well, if you work with little children, you realize that little children don't necessarily do what you ask them to do. Those of you who have any experience of babies, for example, know full well that they don't understand you, and even if they did, they can't control their body sufficiently to maybe execute the task that you would like them to do. So what developmental cognitive neuroscientists do is try to capitalize on the spontaneous behaviors that babies and young children have. So here are 3 different examples. Unfortunately, it's a little bit light, but what we do is capitalize on their spontaneous desire to play with objects and see how the different ways that they play with things can help us understand how they conceptualize the world, how they perceive the world. We might, over here on the right, we have a study with some newborn babies. We might take advantage of the fact that very little babies, newborn babies, prefer to look at novel and interesting things. So by assessing what they prefer to look at, it gives us an idea of what they think is new, what they think is not new, what they think are similar, what they think are not similar. And here we have a school-aged little boy. Now, this is a very difficult picture to see, but what we're doing is really using new technologies here. This little boy engaged in a video game with 3D glasses. Now, of course, he's looking forward, so you can see the back of his head, and you can't see any 3D because this picture is not in 3D, but he's wearing special goggles that give him a real sense of depth in it. And the point here is that new technologies are very captivating to children. Children in the school age will love to spend hours and hours, I hesitate to say, taking part in our studies because they're captivated by these new kinds of technologies. So again, we have to try to find a way to get the children involved and the babies involved because they don't really follow what we ask them to do otherwise. So a second dimension in developmental cognitive neuroscience is to take the biological constraints seriously in trying to understand what is going on during child development. So the first aspect of this is the physical growth. Of course, when we see a child develop, the first thing we notice is that it grows in size and in strength. And in fact, this has a huge impact on the kinds of experiences the child can have. Growth in strength of the leg muscles, for example, enable the child to start walking and to explore his or her environment more independently. And this is often ignored in more standard approaches to understanding child development. A second aspect of the biological constraint is really what's going on in the brain over and above the neurons. The neurons are a network of cells, of course involved in reflection and processing information, but this network of cells actually is bathing in a sea of chemicals, in a sea of neurotransmitters such as serotonin and dopamine, and in a sea of hormones such as testosterone and cortisol, all of which impact on how the brain processes information, and impact, for example, on the plasticity of the brain, on how willing it is, how quickly it can take in information and adjust its structure. Of course, it's very hard to know exactly what's happening in the brain when children are engaged in a particular task, but there are a range of new neuroimaging methods that give us some insight into what is going on. So the three most common ones used with babies are, and young children, are functional magnetic resonance imaging, fMRI, EEG, electrophysiology, and optical neuroimaging. The one in the middle, electrophysiology, is really measuring changes in electrical activity in the brain as the child is engaged in a task. So it's more or less measuring direct activity of the neurons, although the actual neurons, the signal is so small that what you're picking up with the EEG is really collections of thousands of neurons working together. So we can find out when neurons are active, but at quite a large scale. The other two, so this is an example of a baby in an EEG type task. They're wearing a kind of funny hairnet thing of sensors, which are entirely non-intrusive, and are just simply passively recording any electrical activity that's happening under the skull while the baby is watching, say, a video or a cartoon or pictures of its mother and so forth. The other two neuroimaging methods, near-infrared spectroscopy and functional magnetic resonance imaging, work in a more indirect way. What they're both recording are changes in blood flow in the brain in response to certain activity. So this is a much more indirect measure in the sense that there's a built-in assumption. The built-in assumption is that parts of the brain that are working hard, that you really need to concentrate with to solve a particular task, are going to require more oxygen and consequently more blood. And the assumption is, well, if we can pick up that there's more blood in that part of the brain, that must mean that this is the part of the brain that is more engaged in the task than other parts of the brain. Functional magnetic resonance imaging has been around for a long time and is used in clinical context with adults. It's quite difficult to use with children though, because it's very important to stay absolutely still. The participants have to stay still and not move by more than 2 millimeters for the duration of the study, which can sometimes be half an hour, even 40 minutes. Now those of you who know anything about 3 or 4-year-olds will know that they do not stay still for that period of time. So there is really a limit to the youngest age that fMRI can be used to. It's around 6 years of age. The near-infrared spectroscopy shown here being used with a newly born baby doesn't have that problem. But it's a new technology currently being developed in part here and in other world institutions. And without going into too much detail, what this one's actually doing is literally shining a light through the skull into the brain and seeing how much of that light gets absorbed. The theory here is that the more light gets absorbed, the more it means there's blood in that area. Okay? So it literally is shining a light through the skull. I should hesitate to say, or not hesitate to say, I should say, hasten to say, that it's non-invasive as well, in the sense that it's just like having a torch put against your scalp. There's no cutting or opening up of the head in any way. Another method that we can use to try to understand what babies are thinking indirectly is eye tracking. Now, eye tracking uses sophisticated infrared cameras to pinpoint where exactly a person's eyes are fixating in space. So, for example, if we give the baby some video image of a ball bouncing or of someone interacting with a toy, we can see what part of the image the baby is focusing on. Are they focusing on the bright red parts, or maybe they're focusing on what the hand is doing, which is functionally the most important thing to be looking at. Okay, and finally, the kind of third method that gets used in developmental cognitive neuroscience is one that you might be a little bit less familiar with if you've done any reading in psychology, and that is the building of computer models. So computer models are a way of testing whether our mechanistic theories are valid or not. If we have a theory, if we think we understand what the mechanisms are, how things interact with each other, be it neurons or be it pieces of information, if we're abstracting at a different level. If we think we understand it and we can describe those interactions accurately enough, we can give a list of instructions to a computer program, this is a simulation, and tell it to follow those interactions, to mimic those interactions. If then the computer program mimics the behavior that we observe in children, or mimics the kind of brain blood flow that we see in the brain, then we can be pretty confident that we've characterized what's going on in the brain or in the child quite well. If, on the other hand, the computer program does not behave in any way that is similar to the child or the brain, then it makes us think again about whether we're missing some crucial piece of information, whether our theory is complete and adequate or not. So computer programs are a way of validating, testing mechanistic theories we might have about what is going on in the brain. They're used in much of the physical and biological sciences, and have only relatively recently been introduced in psychology through this mechanism of developmental cognitive neuroscience. Okay, so that was just a little overview, as I said, of what are the different elements of developmental cognitive neuroscience. And in the module, developmental cognitive neuroscience of typical and atypical developments, you would see how these methods could be used to try to understand why some children have one form of developmental trajectory, one that might be characterized as typical, or maybe a different form of trajectory, one that's characterized as atypical. And that's it. So if you find this interesting, can I encourage you to come and have a closer look? Thank you.